keep your best years ahead. Longevity Roadmap with Dr. Buck Joffrey.
Hey, everybody, it's Buck here with Longevity Roadmap. Really interesting conversation we got today with Dr. Vera Gorbunova. She is an expert, in longevity. She's a researcher, but her focus is interesting because she kind of does this comparative biology thing, which is really cool because, you have all of these species that have related genetics. I mean, I think we're like, 90% related to mice, things like that. So it's really interesting to use those differences to try to determine why it is that certain species live longer than others. And that is a very, very interesting way to approach this issue of longevity. So hope you enjoyed this conversation. I certainly did. And let me just take a minute also to remind you that if you haven't done so, if you go to longevityroadmap.com, you will find my coaching program. It's effectively a video course and coaching program. I think if you are serious about trying to optimize your health, doing it in a very scientific way, I'm your guy. Now, certainly there are other approaches to this. I've seen others get into, functional medicine and, chiropractic and all that. If you're into that kind of thing, that's not what I'm doing. It is a very, very much a data-driven, science-driven type thing. No voodoo here. So anyway, check it out, longevityroadmap.com. And with that, here's a show. Welcome back to the show, everyone. Today, we have the pleasure of welcoming Dr. Vera Gorbunova, a distinguished professor of biology and medicine at the University of Rochester and co-director of the Rochester Aging Research Center. Dr. Gorbunova is a pioneering figure in the field of aging research, particularly known for her innovative work in comparative biology and the study of exceptionally long-lived mammals. Her research focuses on understanding the mechanisms of longevity, genome stability, and cancer resistance in animals such as the naked mole rat, bowhead whales. Her work has earned her numerous prestigious awards, the Cazarelli Prize from the National Academy of Sciences, the Prince Hitachi Prize in Comparative Oncology, and recognition from the National Institutes of Health. Welcome to the show, Dr. Gorbunova.
Oh, thank you.
So why don't we just start with this? How did you end up becoming so interested in the idea of aging science?
Well, I think aging is probably the biggest biomedical problem of humanity and
Right.
also social problem. So I was interested in basic biology, but then, what other problem is more fascinating because here there is a fundamental scientific problem because everything ages and how does this happen? And then it is so important for human beings.
Yeah, absolutely. So you use what's called a comparative biology approach. So you're studying species with different lifespans, hopefully identifying unique longevity mechanisms. Can you explain how comparing these species really drives innovation in aging research and why it's so important for human implications later?
Yeah, so there are different approaches people can take to study longevity. Traditionally, people analyzed model organisms, for example, fruit flies that have very short lifespan, and then you can manipulate them and you can then select flies that live longer, which is very helpful. But then, because you start from a creature that is so short lived, um, there may be a limit to what you can find, really.
Sure.
And in nature, we find species that are extremely long lived, and there are some animals that live longer than humans. So, for me, the motivation was to find out how evolution solved that problem. And then, if we can learn from that, there is also a higher chance that whatever human intervention we come up with will be beneficial and will not compromise us anyway, because you can in biological system everything is balanced, and if you tweak something there is always a danger that something else will be negatively impacted. But
Sure.
then if we take from nature, like for example there is an animal that lives 200 years, if we use that mechanism it's more likely that it has already been tested and it will not have those negative effects.
Yes. Yes, understood. Yeah, I want to use an example just to sort of help the audience understand kind of, how your research with different animals can work. So your team made headlines by transferring naked mole rat genes into mice, and that led to improved health and lifespan. So maybe you could talk about why this kind of translational work is so pivotal and how insights from these long-lived species can actually potentially in the future may be applied to human aging using that example.
Right. So naked morat is a very interesting rodent. They're the same size as mice, but they live more than 10 times longer. And we researched naked morats, and then we found a gene that is very important for their cancer resistance at that time. And we hypothesized it's important for longevity, but we didn't know unless we could test. And of course, with naked morats, it would take a very long time to test because they live 40 years and more.
Oh, wow.
But when we transferred that gene into mice, we saw that mice, first of all, they had less cancer now, but second, they started living longer and they were healthier. So that was very exciting. We understood that changing this, tweaking this pathway reduces cancer, makes animals healthy, and allows them to live longer. So now, of course, the next step, how do we go now to humans? And because now we understand the pathway a lot better, we can try to come up with some medical ways, sort of pharmacological ways, because we don't want to engineer genes into people, but we can find pharmacological ways to tweak the same path.
Yeah, just to kinda emphasize that study, can you tell us a little bit more? I mean, for one thing, I mean, mice, rodents, they typically die of cancer, right? I mean, that is what they die of typically. So, what kind of benefit did you first of all, what genes did you transfer? What kind of specific effect did you see?
So the gene that we transfer is called hyaluronan synthase 2, which is a gene responsible for the synthesis of hyaluronan. And many people may be familiar with this. It's this polysaccharide that is found in cosmetics. People like to put it on their
Yeah,
skin
yeah,
because
hyaluronic acid, right?
yeah,
Is yeah,
hyaluronic acid. It is very good for moisturizer, but we also have it inside our bodies. It fills spaces between the cells. Uh, so it is a very important component of our tissue, but what we found that in the naked mora, they have 10 times more of it.
huh,
And also the structure is slightly different because these long sugar chains in the morats are way longer than in humans, which makes the molecule more viscous and changes sort of biophysical characteristics of the tissues as well. So this is what we transferred, this one gene.
yeah.
And amazingly, mice started to have much less inflammation as they get older, because inflammation is very important driver of many age-related diseases.
Yeah.
And reducing inflammation was very critical. And now we see the mice are less inflamed. They live longer. Their physical performance was improved in old age.
Interesting. I guess the other thing, so we talked about inflammation. I was about to say inflammaging, which is kind
Yes,
of the the
that's
the,
great.
the concept. So you're finding, hyaluronic acid helps with that. I'm just talking about maybe some of the other things you're finding. You've highlighted the importance of DNA repair and longevity as well. How do the repair processes differ between short-lived and long-lived species? Maybe you can talk a little bit about that, just kind of maybe give us a little bit of a general overview so people, if they don't understand what we're specifically talking about there, but with DNA repair, some of the major mechanisms involved and how those might be different.
Yes, so we are studying different animals. We don't limit the research to naked morad, which is an exceptional animal. But there is another. Well, there are two strategies we can study particularly exceptional species and just delve deeper into their biology and then see what we can learn from them. Or we can also compare maybe 50, 100 species of animals and see what is common among those that are longer lived. And what we found was that DNA repair
Yeah.
is a very big one. Long-lived species really have much better repair of DNA, and of particular type, actually. It is called double strand break repair. This is when both strands of DNA molecule are broken, and that repair mechanism seems to be extremely important for long life. So we find it repeatedly, and we also looked into what enzymes and genes may be responsible. So one that we gene that is very important that we identified to be kind of more highly active in long-lived species is called sirtuin 6.
Okay. Yeah. Yeah. Sure. And maybe you can talk a little bit about what sirtuin 6 does. A lot of people in the longevity space have heard of sirtuins because of David Sinclair's book. And he talks about sirtuins. But what exactly do sirtuins do?
Well, they do many different things. The very original sirtuin, sirtu, that was found in yeast,
Yeah.
was important for maintaining sort of organization of the DNA within the nucleus and for keeping certain parts of the genome silent, so sort of the silencing factor. And when yeast would lose the silencing, they were aging. Now, in humans, in mammals, there are many genes within that one family that corresponds to original yeast gene, and they assume different functions. But sirtuin 6 that we study may be one of the perhaps closest to the original function because it is responsible for maintaining silent chromatin and organization of the DNA within the cell, within the nucleus.
What kind of differences do you see in SIRT6 expression? And, say, for example, I don't know if you're, in this case, if you're looking at the bowhead whales, like what, if you look at, these species that live longer than us, maybe if you could give us some sense of like, how much more magnified their expression of SIRT2, like SIRT6 would be compared to us or, a species that doesn't live as long.
Well, we find high expression, but what that study primarily focused on is the other properties of the protein itself. So sirtuin 6 from long-lived species, like as an enzyme, because it can mediate some enzymatic reaction, was more active, maybe up to 10 times more active, depending which species we compare. So that was quite remarkable that the protein activity has evolved to be higher in long-lived animals.
Are you saying that that's not just a function of having more SIRT6, more expression of the gene? There's some reason, some other mechanism that is more active in longer lived
Yes,
species?
yes. So in that study, we actually first um, just expressed these different enzymes in the same cell line, like human cell line or mouse cell line. And these different proteins were expressed at the same level. But when we asked them to catalyze enzymatic reactions, we saw a very big difference. And then we could find specific changes within the protein that was making it more active.
Okay, so there is some difference in the actual protein itself that makes it more active.
Yes, yes.
Interesting. Okay, got it. So obviously you talked a little bit about sort of DNA repair in general. I just want to make sure we mention a few other things that you found. There's also some things related to what are called transposable elements. These basically they're kind of like jumping genes, right? DNA sequences that can move around the genome and potentially cause damage. And so how is their activity regulated differently in long-lived species? And what might that mean for, humans and later as well?
Yeah, this is a very fascinating topic. We haven't really studied transposons enough cross species, and there are reasons for that because this is so-called junk DNA. And even though we have genomes of many species, but usually the genomes cover very nicely the functional part of the genome, and the annotation of transposons is lagging behind. So most of the knowledge, so even though I studying comparative biology, but we also using other approaches like focusing on human or mouse in greater depth. So most of our work on transposons is limited to human and mouse. But what we found is that with aging, um, transposons start to get activated.
Mm.
And that triggers inflammation because when they are activated, they start to make a lot of RNA, and then they also reverse transcribe the RNA into DNA. And some of that DNA is found in the cytoplasm of the cell where you shouldn't have any DNA. And if the cell finds DNA in the cytoplasm, it thinks that it triggers viral response pathway. So it triggers inflammatory cascades. The cell thinks it's infected. And this is what happens as we age. Our cells start to respond to our endogenous parasite
Yeah.
as if it's an invading virus, which triggers interferon release, other pro-inflammatory pathways.
Do we have an idea of why there's more of these transposons creating problems as we age, and what the
Well,
reason for
we
that is?
understand some of it, but not all of it. So it has to do with this loss of epigenetic structure because our DNA has to be packaged in a certain way within the nucleus, and of course, over time, DNA has to be replicated, transcribed. It needs to be open and closed again, and it's difficult to put
Yeah.
things back exactly the same way they were. So there is some loss of information over time. So there may be this passive, just stochastic loss, which results into the loss of repression on transposons. There is other evidence from our group and others that there may also be some active process because transposons are parasites, so they are trying to free themselves from suppression, and they may be succeeding with it, especially as we get older and our
Huh.
other defense mechanisms start to wane. Um, by the way, we talked about sirtuin-6, so one of its role is suppressing transposons
Okay.
and also DNA repair. But as we get older, there may not be enough SIRT6 to cover all the bases because it has to attend to damaged DNA sites, it has to bind to transposons, and it becomes limiting.
Yeah, you mentioned epigenetic changes. Epigenetics, just as a reminder for people, essentially involves chemical modifications to DNA without changing the sequence of the DNA that ultimately affect how genes are expressed. And so, you may have heard this as a loss of information or something like that over time. So, how do these epigenetic patterns differ between the shorter lived and longer lived species? I mean, is it, there's a lot of epigenetic clocks even that humans are using now. So there's some perspective there. Are you seeing the rate of epigenetic aging much slower in these longer lived species like the whale?
Yes, absolutely. The rate of epigenetic aging really scales up with species maximum lifespan. And a very important question is to understand how they manage to do that. Like because if you think about stochastic process, you would think, oh, it should happen at the same rate for everyone, but it doesn't, which most likely reflects that longer lived species have better maintenance mechanisms that can prevent this disorder from setting in, and we study it very actively, trying to find and then how can we tweak those mechanisms.
Yeah. Do you think that goes back to the SIRT6 expression again?
Yes, so I believe that CERT6 plays important role here, and that possibly one strategy to deal with disorder is to express more or to activate sirtuin-6, and then in old age that may help um, sort of stabilize the epigenome and maybe rejuvenate it, return it back to more youthful state.
Yeah. I want to switch over to mitochondrial function. So people remember the mitochondria ultimately, so we call it the powerhouses of the cell. They're really central to energy production and cellular health. So, how do mitochondrial dynamics vary across species with different lifespans? One of the issues with mitochondria that I talk about on the show is that, I mean, we need them, right? But they also create oxidative damage. And it's a very challenging situation because it's like the machine. I think Aubrey de Grey talks about this in terms of everyday wear and tear. If you're going to use this machine, the mitochondria are going to inherently get damaged. What are you seeing across species there that maybe in the longer lived species, they are somehow better adapted to have less mitochondrial damage?
Yes, so there are different strategies that we see. Some species have indeed more accurate mitochondria that releases fewer free radicals. Other species may just upregulate defenses against free radicals. So there are lots of strategies here. Birds are very interesting in this regard because when they fly, their bioenergetics get so high and they need to deal with it. So, in general, they have more accurate mitochondria and very high levels of antioxidant enzymes.
Yeah. Are you looking at anything specific to mitochondria in your lab? I mean, there's some things that I hear that are interesting, such as mitochondrial transfers. Migration of mitochondria is something that I've just started hearing about, which is really interesting. Any of those kind of concepts?
No, not at the moment. We worked with mitochondria a little bit in the
Yeah.
past, but now we are focusing on the nucleus for the time being, although, you
Yeah.
never know where science leads you.
Yeah. I think I guess the moral of the story for me is that there are a number of these sort of changes that you see, and there's clearly changes or differences between these longer lived species and us. And I think it reminds me, I was interviewing a gentleman by the name of Charlie Brenner. I don't know if you know who Charlie Brenner
Yes, I
is.
know you.
Charlie, Charlie's a really smart guy, and he's the guy who invented Niagen, one of the NAD, the guys who really discovered the NAD pathways and the links to sirtuins, in fact, as well. And he's also what I would call a major skeptic of longevity, of increasing lifespan and such. And I did ask him one question that I think is useful in the context of what you're doing, which is the big picture here is that it does seem that based on comparative biology, that the answer or the solution to living longer lies somewhere within the expression of the genes. Like there's something different, some genetic element, some probably more than one, that distinguish why bowhead whale lives as much longer than a human being. Do you foresee some of the other technology that we have out there, for example, CRISPR technology, to one day be able to find a gene that makes a substantial difference? Maybe it's a cert6 or something like that, and use that to essentially use in humans and therefore improve our ability to combat aging the way a bowhead whale might.
Yes, of course, we would like to get there eventually. Um, I don't know if one gene would be sufficient because to get substantial increase in lifespan, well, in evolution, we get this hundredfold differences between a mouse, for example, and bowhead whale. Um, so to get this kind of jump, evolution changed many things. So I think more likely we would have to use some combinatorial approach to target a few pathways.
Yeah.
And of course, if we could come up with a safe way of doing so, and I think we will, it's just a matter of time, of finding a safe technology and maybe engineer some changes like in sirtuin 6 to make it more active and some other enzymes. Then yeah, maybe we could achieve that significant lifespan extension.
Is that your vision? I mean, it would seem to me that if I'm you, that's kind of what I'm thinking here. But what's your big vision here? of where this all ends up?
Yeah, so I think at the moment there are still some challenges in gene therapies. So I'm primarily, for the near future, we are thinking more about pharmacological approaches, but I think we have to be ambitious. So eventually we may be able to do that, to engineer some uh, genetic changes that are found in long-lived species or even in long-lived humans, because, well, there are humans who live to 100 and beyond, and they have unique genetic compositions. Again, people fail to find a single gene. Again, it's a combination of different alleles, but we can learn from both long-lived humans, long-lived animals, and then we need a very efficient and safe gene editing system to allow everyone to enjoy these
Yeah.
gene changes, because otherwise we say, okay, someone is lucky to inherit from their parents um, this combination of genes, but can we make everyone else
Yeah, yeah,
benefit?
sure. So you mentioned that your focus maybe right now is on pharmacologic intervention. What are some of the agents or supplements or whatever that you think actually may provide some impact for people?
Well, so here again, it's very important to recommend something that is safe,
Sure.
because unlike treating a disease, like here we're suggesting something for people to just to make them healthier.
Yeah.
So
Maybe a
some
better question is, what are you working on that you're excited about?
Well, I'm very excited right now on sirtuin activator called fucoidan. And this is
For
a polysac
coiden? For
fucoidan.
fucoiden. Okay.
Yes,
What?
from the word fucose. It's a monomeric sugar, but fucoidan is a polysaccharide of many units of fucose and maybe a few other like galactose mixed in. And it is mainly derived from brown algae. And what's amazing is that brown algae is a very popular food item in Japan and South Korea. And these are two countries where people have the longest life expectancies.
Oh, interesting. And
So
what does it do?
it is a very strong activator of sirtuin 6.
Interesting. And how does it do that? Is it similar to, NAD pathways, or is it
No,
very
it
different?
doesn't work through an AD pathway. It just directly works on SIRT6. So the exact biochemistry we are still figuring out.
Yeah,
But in vitro, we can take purified sirtuin 6, mix it with fucoidan and a substrate, and we see that enzymatic reaction sort of output increases up to tenfold.
really? Wow.
So it really changes how sirtuin 6 works as an enzyme.
Have you used it in the animal? What kind of results have you seen in the animal?
Well, so we have a manuscript that will soon be, posting, where we see that it extends lifespan of mice.
Mice. Very good. Well, great. How can we learn more about your work, Dr. Gorbunova?
Well, it's mostly my website,
Uh-huh.
Gorbunova Lab.
Yeah.
You can read about the projects.
Fantastic. Thank you so much for your time. It was a very interesting conversation.
Well, thank you. My pleasure.
Thanks for listening. A quick reminder that while I am, in fact, a surgeon, nothing I say should be construed as medical advice. Now, make sure to include your physician in any medical decisions you make. And also, if you're enjoying the show, please make sure to show your support with a like, share, or subscribe.